Thank you, Deborah. Since we're doing great on time, if you have any burning questions about the hollow fiber presentation, please ask them now. And if not, I'd like to invite, if you can stay here for the panel. If I can invite all the speakers and Paolo and Jerry and Tawanda to join me here for the panel discussion. Thanks for finding that extra chair. Okay, so in terms of the discussion, you saw kind of a broad spectrum of quantitative potential solutions to inform decision-making in drug development. You saw something that could be applied across the entire drug development spectrum in terms of the systems pharmacology work that Natasha presented. You saw some things that could be applied to optimize decision-making when you're transferring from phase 2 to phase 3, and then you saw the hollow fiber system work, which allows you to bridge the translational gap between the preclinical and clinical stages. So I guess the first question I would like to pose to the group, and you can each panelist can give it their own perspective on this, is now that those tools are reaching maturity, and we saw an example of something that is already being applied to inform drug development decisions. What do you think are potential hurdles that we should think about, and how to solve them to make sure that these tools are adopted and applied? So, Tawanda, you want to start?
Sure. Thank you very much, and actually want to thank all the speakers. So the hurdles, so here's how I think about it. You know, so you have a model, you have the hollow fiber model, you have in vivo models, and we do talk and work with Eric Nürnberger over at Hopkins. So these are all, so the way to think about it is a toolkit, all right? So it's a toolkit of several tools, and I absolutely love the quantitative and simulation tools. It's sort of actually my first love. Not the hollow fiber, yes, I like, but you know, I love that part. So it's bringing them together, and even from the discussion this morning, the question actually is going to be on further tools to translate. Now these are all translation tools. Now we should be able to map. If we see this in this model, it means that in clinical trials, right? So that's where we need, right? So that we can give people who are up here up front, so that when we develop these regimens, especially when you start talking about shortening therapy and all those aspects, how do we map it, right, to where we can say to the clinical trialists, you know, we did this in the hollow fiber, we did this in the mouse, we did this, and this is how much dose and what combo you have to give for this long to have a 2-month regimen, right? So that's the, so we'll need to be able to map, and there are mathematical ways to map it. So there's forecasting we did when we're looking at the regulatory work, how much do they forecast quantitatively, but I think we need to go a step further and say, no, what is it that we hand over to clinical trials, right? You know, what dose do we give to Dr. Phillips or other people who are up here? What dose in combination with what other dose for how long and how many times a day? And tell them this will give you 98% cure. So there's that gap that, you know, we still need to we still need that granularity of translation.
Jerry, as a drug developer?
I think the biggest hurdle is credibility. increased credibility will come only with increased experience. Each of these presentations, very nice presentations, gave examples of validations, and indeed the last presentation was essentially all about validation. And can one imagine a more in-depth validation than what they did, and then be told, okay, this is a good tool, it can be one among many as a complementary tool to go along with every other source of input you might have in your decision making. But maybe ultimately that's the best that one can hope for, that models are useful for making predictions under certain assumptions, but if people question the assumptions, then they will question the predictions, the inferences. And so it has to be considered as a part of a body of evidence that includes evidence to support the assumptions, includes past experience with the models in making successful predictions, and maybe some confidence in the modeler. If you believe, because indeed for some of these things, like the quantitative systems pharmacology model, it's going to be the very rare individual who actually looks at the code and studies the equations, makes sure that, first of all, that the theory is believable, and then second of all, that that the theory was properly implemented into the computer code. So I think, again, to summarize in one word, it's credibility, which I hope will come over time. I think this is all good work, and it just has to be shown that it repeatedly succeeds in making good predictions.
Do you want to go next, Deborah?
Yeah, if I can, just so I don't lose my train of thought. So we're talking about the validation work, and I think I've shown just a snapshot of what's gone into it. A lot of time, a lot of funding has gone into the retrospective assessment of this particular tool, which people are already using to make decisions. I guess what I'd like to remind this group is I know that there are several new models being developed, tools like the Kramnik mouse. It would be nice if we are prospectively gathering the kinds of data that would be needed to validate a context of use for these specific tools, if the intention is to use those methods for drug development decision making.
Yeah, thank you. Nastya, from your perspective as an employee of a CRO that essentially develops these kinds of tools for clients, what do you see as hurdles and how to overcome them in terms of the application?
Yeah, the main problem, is, the modeling, and, as everyone is saying, is the validation of these models and coming up with something that's acceptable by everyone so people can use it. For our projects, we are trying to translate this in a platform where people can use their own data to compare treatments, something they have, to all the treatments that already exist. We will be showing this platform like this. It's in early phases. We'll be showing this on Wednesday. But we are also hoping to include more options in this to allow people to simulate. So for to predict for different dosing regimens and different durations of treatments to allow them to take some decisions, based on these simulations. Yeah.
Thank you. Now, Natasha, you coming from the systems pharmacology world, that's where a lot of people get worried with the number of parameters in your models, yes, and those kinds of things. So if you could comment on that a little bit, that in terms of the challenge for application, that would be great.
Yeah, I think in modeling we always try and simplify things as much as possible, and systems pharmacology is all about adding as much complexity as we possibly can. And I think to echo what other people are saying, validation then becomes a real issue because you don't necessarily know what impact every parameter has. Is it really necessary? And that's why it's so important for us to simplify our complex model again, to reduce this as much as possible. But definitely validation is one of the biggest barriers we face. And arguably with models, you can never validate a model. You can only make sure that you can't invalidate it. And that's ultimately the only way forward, is to throw everything we have at it and see what our outcome is. Yeah.
Aldo, wrap it up as somebody who
Yeah.
has seen this almost all.
Well, yeah, I'm going to repeat some of it because, I mean, the biggest hurdle is credibility and the fact that people are skeptical. And I mean, I'm obviously a modeler, so I'm biased. But if it's true that, you know, the devil is in the detail, and it's true that one has to check that everything is actually done properly, and this is very complex, and how can you be sure that this is actually, you know, modeling the real phenomenon? Well, I would actually ask the other question. If the system is so complex, how else would you go about it? If it's so complex, would you rather, like, you know, have a thumbsuck guess? Or, you know, you can't run a new trial every time. So it is complex. But people should see these models as something alive, as opposed to, you know, we've done this analysis, now we press play, and we're going to have all the... It's, you know, the whole point is actually finding the failures of the models, the part that don't work, and then collect more information, and then include it in. But the idea is to have a platform, as opposed to an oracle, you know? It's something where you can plug in additional information. So if you study something in the hollow fiber and you see some antagonism between two drugs, it's hard to see how that would translate directly. So you use the model to ask that question. And if it doesn't work, well, it means that you need to refine it further. I think that's it's the concept.
Tawanda?
So one of the things that's very difficult to do, it's probably only in academia, I'm joking, but I know in pharma, is, so when you have your tool or your model or your molecule, right? So it's hard to see your own blind spots, right? So, because all models have limitations, right? And we usually don't think of what our models can't do, but it's very important to think of what our models can't do, right? Otherwise, we promise the world to people. So one way, I think, and Deborah alluded to it, which was with hollow fiber work, and now going on to the mouse model, is to at least start giving people parameters of how accurate is forecasting with these models, a systems pharmacology model, knowing that they will continue growing and changing, of course. But let's start giving people metrics on how well this does, right? Because that also tells us how well they don't do and what they don't do. And if they do well, what aspect of translation do they do well in? And then we go back and either work on expanding to those things they can't do, or look for another model that addresses what this particular model doesn't do. We don't think of it because, you know, so you hear a lot, and I'm not going to say names, this model, this animal, and so move towards, well, what does this animal do? What does this hollow fiber do? How accurate? And if not accurate, why isn't it accurate? What can't it do? And I think, you know, platforms like CPTR and certainly the foundation, the Gates Foundation and so forth, give that, I hope, platform where we can, so exactly as was done this morning. Why didn't it work? Why did all these treatment shortening studies not work? What was done wrong? What can't we do with what we used that we thought we were doing and got it wrong?
Yeah, and actually that brings up an interesting point, because if we're to embrace this notion of learn and confirm and understand really what a model cannot do, part of that conversation also has to do with, are we having gaps in the data that are not letting a particular model really achieve the level of predictive accuracy that we would like? So that's also, I think, an opportunity to have that conversation back with the experimentalists, both preclinically and clinically, to inform the kinds of data that would actually help us refine those models and keep them alive and keep them growing. And probably one model that may not necessarily be as great become a useful model just because we fill in those gaps for the data required. So I wonder now. I'm just going to pick a little bit on you, Jerry. If you can give your perspective as a coming from the drug development world, how you potentially do you really see the value in embracing that concept and having that information as opposed to blindly trying to figure out what data to collect?
I think that's a great point, if I understand it correctly. So you're saying the model is actually a guide as to what data to collect to help you further inform the model. Okay, yeah, I think that's a very nice insight, and that's frequently a challenge to a modeler. Is to be able to have enough influence to be able to get the proper experiments conducted, or to be able to get the data, sufficient data that you would like collected in a clinical trial. But again, as the models prove their utility, then I would hope that we can build a kind of community of appreciation and understanding in what the models can do that then justify the expenditure of the extra effort to get that data, and that everybody's understanding grows because of that. It's a good point.
Awesome. Thank you. Paula, do you want to comment on that a little bit, that learn and confirm paradigm?
Okay, yeah, I agree that I think what the model can or cannot do, I mean, sometimes it's just help seeing what the sort of limiting factors are. I'm thinking, the very complex model that Natasha described. I mean, where does it seem like the bottleneck is? Is it, penetration into the lesions? Is it allows to separate something so complex and sort of focus on particular aspects and say, okay, it seems like this is the issue. Is there data out there that can answer this question already? Can we design an experiment to respond to this? Because since it's, when you run a clinical trial, all the elements are in the picture. But that, but on the other hand, the clinical trial is the only one that is going to tell you whether people are going to get cured or not. If instead you just run a specific experiment, then unless you have a framework to plug it in, the experiment is only going to answer a very small question, and you don't know how that fits in the picture. So I see the model as something, a platform that can help you, plugging in single information and see how that converts into the general picture, and if it doesn't, what you should do to fix it.
Yeah, great. And now, related to that point about embracing an application and implementation of the models, one of the things that we've also faced, not just for the TB community specifically, but across the board, is the notion that these models require you to know how to program in specific languages and whatnot, and that is very off-putting to a lot of people. So I wonder if both Natasha and Nastya could comment a little bit on this, what we've discussed about graphic user interfaces that would allow essentially any member of a preclinical or a clinical development team to make use of the models in a click-ready environment. So.
Yes, so sometimes it's very hard to explain all those parameters that we have in an equation to people with a more clinical background or who are just interested in looking at the shape of the graph without, knowing what's happening in behind. So this, brings us to the importance of these platforms where people can look at different drugs, look at whatever is important for them without, having difficulties understanding those complex parameters. And this is why we're bringing those platforms to allow people to do whatever they need to do, compare drugs, compare dosing regimens, and be able to take their decisions and without seeing all the work that's in the background. Yes, so these platforms are becoming very important to make a bridge between modelers and clinicians.
Yeah, absolutely. And I think to add to that, you're speaking about other people who have frustrations with learning code. I think a lot of this comes from modelers being frustrated that people don't understand what we're doing. And we just want to make it more accessible to a wider audience because we still believe that modeling is fun. We want to make sure that people can see the advantage of it. And obviously having a nice graphical interface where people can try their own drugs and see the potential of the work that we're doing is really exciting.
Great. Yeah, yeah.
It's part of the problem and way of thinking. So if I can put on a clinician's head in the whole field, right? In that, and Klaus is smiling because he probably knows what I'm going to say, but think of it. So we're used to thinking in very binary fashion in the clinic, right? This get better or when the patient get better. So, and part of the problem, and you see it in a lot of fields outside TB, outside infectious diseases, is beginning to think in a very quantitative fashion across spectra, right? Accept 80% response, 90%. What does it mean, right? So I think a lot of the frustration with modelers is we can't explain to people without resorting to quantitation. But a lot of our frustration with them is, why don't they resort to quantitative way of thinking? And so we're behind the economics and the engineering and all those people who already think in that fashion. And so it will necessarily take time. My only major gripe with it is we would hope people in drug development think in a quantitative fashion. And if we can reach those, right, in that way across, and for those who are developing all these models, if we can develop models, not that the average person in a program, TB program can use, but at least that companies would be able to use for quantitative reasoning, then we can declare at least that we've reached some way to be useful.
Yeah, I just want to add one more thing about these interactive tools. They're not just for non-modelers to be able to play with, but one historically inefficient part of modeling has been that a modeler may be sitting at a clinical team meeting and a question comes up that can be answered by a model, but then the modeler has to go back to the office and maybe spend a few days rejiggering the model and then come back with some plots on paper or a PowerPoint presentation or something a week later, and by then the project is moved on and it's inefficient or useless. But now the modeler can sit in the clinical team meeting with a laptop projecting on the screen, and somebody asks a question, and you just change a few things on the slider, like in the interface that we saw, and you get instantaneously an answer in terms of a simulation. So it's a very nice tool for the modeler and thereby for the entire clinical team together to be able to take advantage of the models more.
Yeah, that's actually a very great insight as well as to how that can happen. But getting back to Wanda's point, and you can comment on this if you agree or disagree, and Paolo and others on the panel. But maybe that's the having those GUIs, those graphical user interfaces, maybe the start to get a broader type of audience interested in what's under the hood of those models, because once they get something that they can readily use, so that's a great starting point. But as they start using it and they start generating those graphs and those tables, questions are going to come up, and that's going to prompt a discussion, not just from the model trying to teach the model from the bottom up, pun intended, but more from the user trying to understand what's behind a curve that seems a little odd, or a table that seems a little strange, and that's going to prompt that level of conversation. Would you agree that that could be a way to solve things kind of in a reverse kind of way?
I think to the points that people said in the beginning, that helps with three things, and I'll sort of push it even further, right? And people are talking about credibility, and we're talking about how well do these models do, and the credibility to people doing clinical trials or planning. Let's say the Alliance, I'm just picking on them because they're here, and I know them. So the important thing that should get to them is the models. They should be able to use them to answer the questions they need for their clinical trials. And as you get in more, not accurate, but as your forecasting capability increases, then they will use them more and more. So in other words, I'm agreeing with you. So we have to go to the user, right, and say, we have all these equations in the background, right? When they want to increase the dose of rifampin, if Panacea wants to increase the dose of rifampin to 1,800 milligrams, we should give them a tool where they can say, if I increase the dose, what's going to happen to the outcomes via simulations in these models. Something much more useful to them. And graphic, interfaces are great. I mean, that's what we do with Windows and Word, right? We no longer type all those instructions. So for me, we're partly to blame in not giving people the tools for the questions that are important to them. There are questions that are important to me. I like kill parameters and all that stuff because that's what I do. But they want to know what proportion of patients will respond in 6 months or in this month. So if we can move our tools, so I'm agreeing with you, to where people input the parameters that they want and they can get an idea that's accurate of how well this is going to do, I think we have met at a point where we're useful to each other.
Paula, do you want to add anything to that?
I completely agree that sometimes, the modeler goes through the lengths of developing the whole model, then they put a paper, maybe there's a simulation there, a chart or two, and normally you use a model to answer a question, and then the paper is about the answer to that question. But what is really useful is the model per se, but then if you want to re-implement it, well, sometimes you know the author of the paper, you email them, they send you the code, but it remains sort of in between nerds, let's say. It's like, it's one of those. So the other problem is that, when people work in academia, we all have lots of stuff to do, and very, very often you don't have the skills to implement this sort of graphical user interfaces and things that are user-friendly. So I think there would be lots of value in having like a platform that can host these kind of models. And I don't know if, within CPTR or there could be an initiative like that, because once one knows how to do it, it takes probably about, 1 hour or 2 to implement a model if you already know what you're doing. And that would be like a repository for, the end user to, go browse to the library of models and use them. So I would say that, this would actually go a very long way into converting the research into something that people can use, as opposed to, a chart on the paper that, only answers that question.
So I will certainly say that it's an aspiration of CPTR to develop this platform minimally for the tools that we are building with all the partners that you've heard about as part of our modeling and simulation working group. It's important because it's part of our mission, as you know, to make every tool that we validate or build publicly accessible to everyone for use. So we want to make that as user-friendly as possible. We also are committed to continuing to refine these models. Just like you've seen with the hollow fiber system, we didn't get to an endpoint and just say, good luck, we hope this works out for you, right? There's additional experiments to be done. We want to make sure that we can connect interested developers with a laboratory or laboratories that are skilled enough to implement the laboratory manual and run the studies with reproducibility and validation. It's no different when we're talking about these in silico or mathematical models. So an aspiration going forward to build a platform like that.
Yeah, absolutely. And that actually links to another component that was just discussed, this notion of the continuous validation of the models or the continuous testing to see at what point you invalidate a given model. So it not only ties in with the conversation about looping back with the experimentalists to generate the additional data, but also ties in with this notion that we've largely overcome about data sharing and data integration, which is something that has already been discussed. I just want to conclude with a plug for a breakout session that's going to happen on Wednesday afternoon. Nasya is going to, for those of you who are interested, she's going to show a very user-friendly graphical user interface for the time to positivity model that she showed. And later this afternoon, you're going to hear about another really cool platform, and we're going to have another breakout session tomorrow about that. So with that, thank you very much, and this concludes this session. Lindsay.
First of all, thank you to all of our panelists. And we're going to take a 15-minute break if everyone could come back around 3 p.m. and we'll get started again. Thank you.